Good afternoon. My name is Paweł Urban, and I work in Tsinghua University. Thank you very much for inviting me to this event, and I'm sorry that I cannot be present in person. I will give you a brief overview on our research on skin metabolomics. The main research tool that we use in our lab is mass spectrometry. Mass spectrometry is a very powerful technique. It is characterized with high selectivity, sensitivity, and speed. These three attributes we say triple S. And mass spectrometer consists of three main parts: source, mass analyzer, and detector. And there are some additional components, and from the point of view of user, the interface is very important. How we deliver sample to mass spectrometer, because this can affect the performance of the whole analysis process. And we deal with analysis of unconventional biological matrices. So this is a recent trend in clinical chemistry. So the conventional analysis encompasses analysis of blood, urine, feces, or tissue biopsy, but recently there has been growing interest in analysis of unconventional specimens such as hair, nail, breath, saliva, tears, meibum, sweat, and mucus. And there are various factors that are taken into account when deciding what kind of specimen, what kind of matrix to use in clinical analysis. And we choose skin excretions, and the main component of skin excretion is sweat, and sweat consists of three main fractions. So one is the lipid hydrophobic components, the other cellular debris, bacteria, fungi, and the main component of sweat is our aqueous hydrophilic compounds. And of course this includes water and many inorganic ions, as well as organic compounds, including amino acids. And what is present on the surface of skin is due to different factors. So one is, of course, the secretion of sweat. So sweat is produced by specialized glands that are found in skin, in the dermis layer. Furthermore, apart from sweat, we can also find sebum excretions on the surface of skin, and sebum is produced by sebaceous glands. So moreover, some studies focus on interstitial fluid, so the fluid present in between the skin cells. On the surface of skin, we can also find degradation products of proteins, so some free amino acids that are not from sweat, but they originate from degradation of proteins. Furthermore, when we expose skin to sunlight, there are some photochemical reactions that induce transformations of certain organic molecules to others. Moreover, particulate matter from smoke, from environmental pollution, also can be deposited on skin. Sometimes we apply drugs on the surface of skin, which of course change the chemical composition of the skin surface. And moreover, skin contains microbiota, like viruses, bacteria, and fungi, which also alter chemical composition. So in one of our studies, we focus on skin metabolite analysis in skin disease called psoriasis. So this is an autoimmune disease which affects 2 to 3% of population, and doctors can see some changes on the surface of skin caused by psoriasis, some lesions, and these lesions are characterized with plaque severity score, or PSS, which is due to redness, thickness and scaling which appears on the surface of skin affected by psoriasis. And also there are some systemic diseases that have skin manifestations. So there is interest in detecting skin metabolites which can help to diagnose diseases or to monitor the progression of the diseases. And main analytical methods used for skin metabolomics encompass liquid chromatography, gas chromatography, mass spectrometry, and NMR spectroscopy. And we focus on mass spectrometry, and a few years ago we developed also a method for sampling traces of skin metabolites into hydrogel matrix. So this method uses small probes made of inert polymer, and these probes have cavities which are loaded with agarose hydrogel. Those probes are affixed onto the surface of skin, and metabolites present on the skin are transferred to the hydrogel. Furthermore, those probes are incorporated into special mass spectrometry setup where metabolites can be solubilized, and metabolite extracts can be transferred to the ionization region in the mass spectrometer. So we can see many peaks, and those peaks relate to different skin metabolites. So using this method in cooperation with Dr. Hyun-Yi Cho from the NTU hospital in Hsinchu, we carried out a study in which we studied skin of patients affected by psoriasis, and you can see very good separation of the metabolomes of patient skin and metabolomes of healthy skin, the skin of healthy controls. So these data points in multivariate analysis plots are due to many metabolites that were detected by mass spectrometry in skin excretions. And these are some biomarker candidates that were found in that study. These compounds mainly contribute to the separation of the data points, which you could see in those plots. And here's an example of choline, so a small molecule, and PSS, I just remind you, it's plaque severity score determined by a physician. And this plaque severity score and the normalized intensity of choline present on skin correlate to some extent. So choline is a putative positive psoriasis skin biomarker because its concentration in skin increases as plaque severity score increases. And most recently we try to develop an easy-to-use tool for studying skin metabolomes. We call it mass spectroscope. So basically, this is a probe, a handheld probe, which can be approached to the skin surface. Here it's hand, palm region, and the volatile organic compounds present on skin are immediately transferred to high resolution mass spectrometer, and we can detect them. In some cases we can identify them. So in the case of this method, we do not have to do any sample preparation, we do not have to wait for a long time, we do not have to transfer samples and handle them in any way, because we get the result within minutes or even seconds. And of course there are some other research activities around the world where researchers develop wearable biosensors for health monitoring, but I must say that those biosensors mainly target one or very few metabolites. So that's in contrast to mass spectrometry, because mass spectrometry can detect many metabolites at the same time. And at the end of my short presentation, I would like to highlight some challenges and opportunities for possible cooperation. What is needed, what can be done in the next step. One should focus on discovering and validating skin biomarkers of systemic diseases, because those studies that we do, of course, are done on small numbers of human subjects. Further on, it will be desirable to understand the biochemistry of those skin biomarkers, so their biosynthesis and maybe relations to proteome and genome. Moreover, it would be good to downscale mass spectrometric sample readout. So like the case of our setup, we use quite big mass spectrometers which cannot be placed in the hospital yet, but it would be great if we could downscale this setup so that one can put this mass spectroscope system anywhere. And further on, one has to make effort to transfer the skin sampling analysis methodology to the market, something that we still have to face in the future. And thank you very much for attention.